DNA ligase is very important enzyme to create recombinant or artificial DNA molecules. In addition to DNA ligase, we can use another technique which is called as homopolymer tailing to join DNA from two different sources. तो इसमें हम दो पॉपुलेशंस लेते हैं डीएनए की या सोर्स डीएनए की और इसको ट्रीट करते हैं with certain enzymes. We are going to discuss which type of enzyme. Or this enzyme, it is provided with only one type of nucleotides. Like in one population of the DNA, we have provided only adenine residues. So enzyme will add adenine residues at 3' ends of first population of the DNA. That may be vector molecule. If we consider the second population of the DNA, it can be treated with another single type of nucleotide bases like thymine. So thymine residues will be added at 3' ends of the second population of the DNA, so that single type of nucleotides at 3' end, that may be up to 10 to 40 base pairs of thymine residues. So this stretch of thymine residues, it is called as homopolymer tailing. So when we will mix two populations of the DNA, so because of the presence of adenine homopolymer tail in first population and thymine in another population, so complementation or complementarity it is there, so that two populations they will join together. Now the question is that how we can introduce homopolymeric tails in DNA populations. So this homopolymeric tails can be created by the use of an enzyme which is called as terminal deoxynucleotidyl transferase, और simply हम इसको कह सकते हैं कि terminal transferase. What is the source of this enzyme? It can be isolated from different sources. So one of the source which is most famous, it is calf thymus. So thymus, it is an organ in vertebrates that produce immune cells in the body, in the immune system, so that when pathogenic microbes invade the body, so it can combat and fight with invading pathogens. So this thymus from young cows, it can be a good source of terminal transferase. In addition to calf thymus, it can be isolated from other sources as well. And now it has also been produced by cloning this gene from calf thymus into E. coli. So we can get terminal transferase commercially as well. So what is the mechanism of action of this terminal transferase? When DNA fragments, it is provided with only one type of nucleotides. As I mentioned you that population 1 of DNA, it is supplied with only adenine. So this terminal transferase, it will add adenine residues at 3' end, and in the presence of thymine residues in second DNA population, it will introduce only thymine residues, and length may be 10 to 40 residues. And when these two populations they will be mixed, so recombinant DNA will be produced. So the joining of DNA by homopolymeric tails can be explained with the help of diagram. So here we have two populations of the DNA, first population and the second population. So if DNA population it is treated with lambda based exonuclease, it will expose 3' hydroxyl end when they will be exposed, it will be a good substrate for the enzyme terminal transferase. So if it is provided with only single type of nucleotide, and here it is adenine, so it will add adenine at 3' ends. N means number of nucleotide bases like adenine that has been added by terminal transferase. So this is one of the example of homopolymeric tail where only adenine residues has been added. In second population of DNA, again it can be treated with exonuclease so that 5' hydroxyl end may be exposed, and then terminal transferase, it is provided with only thymine residues so that it will add thymine residue at 3' ends that are complementary with the adenine residues of first population of the DNA. When they are mixed together, annealing it is there because of the complementary homopolymer tails, so that when it is introduced into the host organisms, the nicks that are there between the adjacent nucleotides, it will be repaired by the host machinery. So homopolymer tailing, it is also one of the example by which we can join DNA fragments.